Welcome to this lecture on the fundamentals of biomolecules. Today, we are going to explore the fascinating world of nucleosides and nucleotides. These tiny structures are the very foundation of life itself, responsible for storing your genetic information and powering your cells. Let us dive in and discover how they work. To begin our journey, imagine you are trying to build a massive, complex Lego castle. You cannot just throw a pile of plastic into a mold. You need individual, pre-shaped Lego blocks that snap together perfectly. In the human body, our genetic code, which includes DNA and RNA, is exactly like that massive Lego castle. The individual blocks that snap together to build DNA and RNA are called nucleotides. Before we can understand the whole castle, we must zoom in and look closely at a single block to see how it is constructed. Now that we know they are building blocks, let us look at the anatomy of a nucleotide. A nucleotide is the fundamental monomer of nucleic acids. Structurally, it is composed of three distinct chemical parts. First, we have a nitrogenous base. Second, there is a 5-carbon sugar. And third, there is one or more phosphate groups attached to the sugar. These three components must come together in a very specific arrangement to form a functional nucleotide, ready to be incorporated into a larger genetic structure. You might be wondering about the term nucleoside, which sounds very similar. When you take away the phosphate group and only look at the nitrogenous base attached to the sugar, that two-part structure is formally defined as a nucleoside. Therefore, you can think of a nucleotide simply as a nucleoside that has been phosphorylated, meaning a phosphate group has been added. This modular nature is incredibly important for how cells build and modify these molecules, depending on what the body needs at any given moment. Let us look at a visual representation to make this crystal clear. On the screen, you can see the distinct parts we just discussed. Notice the nitrogenous base connected to the pentose sugar. This connection is called a glycosidic bond, and together, they form the nucleoside, outlined by the dashed box. Then, look at the phosphate group attached to the sugar via a phosphoester bond. Once that phosphate is attached, the entire three-part structure becomes a complete nucleotide. This diagram perfectly captures the modular, block-like nature of these molecules. Beyond just being structural blocks, why do we care so much about these molecules? Without them, life as we know it would be absolutely impossible. They form the alphabet of our genetic code, storing the instructions needed to build every single protein in your body. Beyond genetics, specific nucleotides act as the primary energy currency of the cell, powering everything from your heartbeat to your thoughts. Furthermore, synthetic versions of these molecules are widely used in modern medicine as powerful antiviral and anti-cancer drugs to stop diseases from multiplying. Now that we understand their biological role, let us look at how they are built chemically. To truly understand how these molecules come together, we can look at the mathematics of their formation through condensation reactions. When a base, a sugar, and a phosphate group join together to form a nucleotide, water molecules are released. This means we can express the total molecular mass of a newly formed nucleotide using a simple algebraic equation. This equation helps us track the conservation of mass during the chemical synthesis of these vital molecules. This chemical process can be expressed mathematically with the mass condensation formula. The formula is M equals B plus S plus P minus 2W. Let us break down these symbols. M represents the final molecular mass of the complete nucleotide. B, S, and P represent the individual masses of the free base, sugar, and phosphate group. W represents the mass of a single water molecule, which is exactly 18 grams per mole. We subtract 2W because forming a nucleotide requires two separate condensation reactions, releasing two water molecules. If the mass of the base or sugar increases, the total mass M naturally increases. Let us put this formula into practice with a real molecule, adenosine monophosphate. We are given the mass of adenine as 135, ribose as 150, and phosphate as 98 grams per mole. We need to find the final mass. First, we add the three components together. Then we subtract the mass of two water molecules, which is 2 times 18, or 36. Subtracting 36 from 383 gives us our final answer. The final molecular mass is 347 grams per mole. Biochemists rely on this exact physical value to synthesize DNA in the laboratory. We have successfully built one nucleotide. But DNA is a chain, so let us see what happens when two nucleotides join together. When they connect, they form a strong connection called a phosphodiester bond. Just like the previous bonds, this is a condensation reaction, meaning one molecule of water is lost when the two nucleotides connect. Therefore, our strategy to find the mass of this new chain is to add the masses of the two individual molecules and then subtract the mass of one water molecule. This logic applies every time we add a new link. Just as we calculated the mass of one nucleotide, we can model the mass of this new chain with another equation. The formula is D equals 2m minus W. Here, the variable D represents the mass of the dinucleotide. We multiply the variable m by 2 because we are joining two identical monomers together. We subtract the variable W exactly once because only one new bond is being formed between the two nucleotides, releasing one water molecule. If we were to add a third nucleotide, the mass would increase, but we would again subtract one water molecule for that specific new bond. Let us see this new formula in action by joining two of our previous adenosine monophosphate molecules. We know from our previous example that one molecule has a mass of 347 grams per mole. We multiply this by 2, giving us 694. Then we subtract the mass of one water molecule, which is 18. This gives us a final mass of 676 grams per mole. This physical value tells us exactly how heavy our new two-link polymer chain is, which is crucial for understanding how biological molecules are assembled in nature. This final number leads us to a profound realization, allowing us to prove a fundamental principle of biochemistry. Let us set up the proof. The mass of two separate, unjoined monomers is simply 2 times m. However, the mass of the joined dinucleotide is d, which equals 2m minus w. Because w is a positive value of 18, 2m minus 18 must be strictly less than 2m. Therefore, we have mathematically proven that d is less than 2m. This proves that polymers are always slightly lighter than the sum of their individual free monomers due to the water lost during polymerization. We have covered a lot of ground today, so let us review our key takeaways. First, nucleosides consist of a base and a sugar, while nucleotides add a phosphate group to become the true building blocks of DNA and RNA. Second, These molecules are formed through condensation reactions, which release water and conserve mass predictably. Finally, because of this water loss, polymer chains are always lighter than their individual parts. You now have a solid understanding of the fundamentals of biomolecules.